Hey everyone, welcome back for another Proteomics lecture. So we're going to continue in our discussion of an overall protein purification and characterization scheme here. We're going to do a little bit of a review here. We've already had a in-person lecture on centrifugation concepts and protocols before, but centrifugation is such a key component to any protein purification strategy that we're just going to go ahead and discuss it again here very shortly. So let's go ahead and get into this. So it turns out that a lot of what centrifugation is about is very similar to what we've already talked about with size exclusion chromatography, and we know from that SEC is all about separating proteins on the basis of their size or molecular weight. Okay, so what is it about proteins that would cause them to separate differently on this basis? Well, number one, and the most obvious, is the number of amino acids that protein contains. It should just be fairly obvious that a protein that contains 500 amino acids is going to be a lot bigger and a lot heavier than a protein that only contains 100. And then the second aspect that we've talked about also is, okay, which amino acids are you utilizing? So if you have two different proteins that both contain a total of 100 amino acids, that by no means means that they're both going to have exactly the same molecular weight. We've already seen that glycine is the lightest amino acid and tryptophan is the heaviest. So if you have a protein that is made up of 100 tryptophans, it's going to be a lot heavier than a protein that's made up of 100 glycines. So if you want an example of a very, very, very big protein, the largest known protein, at least as far as human cells go, is called titin. This is a protein that is a major component of sarcomeres within muscle tissue, and it can contain, depending on which isoform you're talking about, it can contain up to 35,000 amino acids. So if you do the mathematical trick that we talked about previously in estimating the molecular weight, if you multiply 35,000 times an average amino acid weight of 110 Daltons, you end up with a molecular weight of roughly 3,850 kilodaltons. That is absolutely massive. And then if you look at the opposite side of the spectrum, the smallest known actual protein that folds into a shape is called TAL, and it only contains 11 amino acids. So you can see here that even though proteins are made of the same stuff, they're all made of amino acids being chained together by peptide bonds. They very much run a very wide spectrum in terms of how big they are and how many amino acids they contain. Now, when it comes to centrifugation, it's not going to be so much the mass we're interested in as much as the density. So mass is important, but we need to also consider density as well, which is, of course, the amount of mass per unit volume. And it turns out that even though proteins very much differ in terms of their mass and how many amino acids they contain, most proteins have a fairly consistent density with an average value of about 1.37 grams per milliliter. So most proteins fall within about 15% of this value, not going much further than maybe 1.3 or so, and not getting much greater than maybe 1.45. Now, the only proteins that are going to be significantly different are those that are either lipoproteins or glycoproteins, in which having a significant extra amount of mass due to carbohydrates or lipids will cause that mass-volume ratio to change. So if you look at this table here, you can actually see some examples of different proteins that have very starkly different molecular weights. You can see an immunoglobulin, which is a very, very, very big very big protein that consists of several different subunits, having a molecular weight of over 1,000 kilodaltons. And then you can actually see a very, very small protein like lysozyme, which we've talked about before, has a very small molecular weight of about 15. So you'll notice that most of the proteins on this list, with the exception of the high-density lipoprotein, have a pretty consistent density, somewhere in the neighborhood of 1.35 to 1.40 grams per cubic centimeter. So they all have fairly consistent densities. So When it comes to centrifugation, we have considered the mass of the sedimenting particle and its density, but ultimately what determines how quickly a particle, including a protein, is going to sediment by applying centrifugal force to it, that is its sedimentation constant, which is given in Svedbergs. So if you actually look at the S value for these different proteins, you'll notice that they actually tend to kind of go down a little bit as the proteins get smaller, with the S value getting a little bit harder to predict the smaller the proteins get. So the immunoglobulin, which is a very big protein, has an S value of somewhere between 18 to 20 Svedbergs, whereas lysozyme has one as low as 2.19. But as I said, this is a little bit hard to predict, and it's not a perfectly linear correlation with the molecular weight. So ultimately, when we're talking about using centrifugation for protein purification, why do it? Well, because centrifugation separates on the basis of how dense a particle is and also how much mass the particle has, it's going to be a critical part of just about any purification scheme. And we've actually already seen several examples of how this can be used. One of the most common ways is right after cell lysis. So once we bust open the cells, we disrupt the cell membrane, and we get all the intracellular contents leaking out, we can centrifuge the lysate to give us a supernatant, which is going to contain all of the soluble proteins, and then you'll also get a pellet that's going to contain all of the insoluble stuff, such as membranes, nucleic acids, and then any insoluble membrane proteins that we did not extract from the membranes if we did not add a detergent. So because we separate the cell into two different phases, the soluble supernatant and the insoluble pellet, depending on what we know about our protein, whether it's soluble or not, we should know which one of those two phases our protein exists in, and we can easily separate the supernatant from the pellet. We've also seen that we can use differential centrifugation to separate organelles from each other after the lysis has occurred. So this is going to be useful if you happen to know that your protein is only located in one specific place inside the cell, such as the case is with something like cytochrome C being located in mitochondria. And if that's the case, if you have that pre-existing knowledge, you can use differential centrifugation or even use a density gradient using something like percol or sucrose to isolate the organelles into a number of different fractions so that you can get a especially concentrated fraction that mostly contains just the organelle that you know your protein exists in, and you can discard all the other organelle fractions that you're pretty sure your protein does not exist in. And then we've also seen, after doing our ammonium sulfate precipitation, that centrifugation is an important step here for largely the same reason as it was in the cell lysis step. After we add a certain amount of ammonium sulfate to get the percent saturation of our solution to the critical point that our protein of interest falls out of solution, we can use centrifugation to pellet it at the bottom of the tube. And once again, we can separate the soluble supernatant away from the insoluble pellet. So again, we've already seen quite a few different examples of how centrifugation is going to be extremely helpful to us in purifying a protein, and it's not necessarily just a one-trick pony. We can use it in a number of different cases. All right, well, that's going to do it for this little review on centrifugation. So join us next time, and we will start talking about something that's a little bit new to us, the concept of gel electrophoresis, especially SDS-PAGE. So thank you for your attention as always, and I will see you next time. So long.